Welcome back. So in this video, I'd like to talk to you about one of the primary things that you're going to do, and that is the sequencing and assembling of a genome. And so this process typically involves, as we talked about a little bit before, taking a perfectly good sample of genomic DNA and breaking it up into a bunch of pieces, and then attempting to informatically, after you sequence those pieces, put it all back together again in something that reflects the original sequence of nucleotides in the genome. And so you'll get to understand why we call this the Humpty Dumpty problem. So looking at this sort of schematically, when we start the sequencing project for each of your genomes, your bacterial genomes that you're sequencing, you're really starting out with a solution that contains many, many copies of your genome in a relatively intact form, long stretches of chromosome, but many, many copies of that. And those are, in one way or another, randomly fragmented. And so, as I told you in an earlier video, we're using a transposase-mediated fragmentation and combined with inserting the adapters for Illumina sequencing into your sequence at the same time. And it generates a set of overlapping fragments. So each part of the genome is presumably represented in this library that we're going to sequence by many fragments that have that originated from different copies of the original genome and have different ends on them. And so those, as part of this, we take those through a process because it's not very efficient to sequence the little tiny fragments. If you think about it, we're trying to generate sequences that are 250 base pairs from each end of the original fragments. And so if the original fragment is less than 250 base pairs, then we're kind of wasting sequences and we'll just be sequencing adapters or out into the middle of space. So we like to keep those as large as possible. But when you're looking at the QC of your sequences, you'll see how close we came to doing a perfect job of that. So what we end up with, if you look up in the top right sector of this diagram, are fragments depicted by each of the bars, and then sequences from the ends of those fragments. And so we know, for example, that the read going in one direction, or R1 as we'll come to call them when you're doing your analysis, which will be 250 bases, is actually paired to another read that's also 250 base pairs that's coming from the other end of that exact same fragment. Okay, and so we can use that information, and intellectually we can use that information just in this diagram to see that, you know, if we could find the sequences and how they overlap, we could make this sort of an overlap pattern here, or a tiling pattern, if you will, that would allow us to put these sequences together into contiguously sequenced regions. And so that contiguous sequence is what gives rise to the name of a contig. And so the primary goal of reassembling these things is to generate what are called contigs. And so the reads, in this case, that you are provided are in the format of FASTQ sequences. And they're paired, so you have a set of things that are read one, FASTQ sequences, and then there's an exactly, essentially matching set that are R2s. So for every R1, you have an R2. And then those are put together, and this is one of not the first thing, but one of the first things that you'll do with your sequences is to assemble them into contigs. And we'll talk about the logic of that and how that actually works in a moment. Okay. Now there are places where either for a whole variety of reasons that we'll talk about in a moment, but even for just because there's a sequence in there that's not represented, or for some reason won't allow you to unambiguously put them back together again, that make you go from one long chromosome sequence, or for many of your bacteria, you would expect a single circular sequence that you started with, and so, but leaves you with many, many contigs of various sizes, okay, with gaps in between them. Okay, and so there are mechanisms, we won't talk about them too much in this context, of using other information to put the contigs together into what are called scaffolds. So I want to talk about this, without getting too super technical about this, about what actually takes place, because it's important, again, in terms of understanding the way these algorithms that are going to build these assemblies are going to work. And so this is a very simplified version of this. And so the general approach that are used for these so-called next generation sequencing assembler is what's called a de Bruijn graph. Okay, and what they're primarily looking for is they take your sequence, your data, your FASTQ files, and those FASTQ files are broken up into what are called KMers. KMers are simply DNA sequences of a particular length. And so in our silly example here, we have taken a sequence and broken it up into KMers that are 3 nucleotides long. Now, of course, we're only trying to build a genome that is very small, and so that's not a problem for us. Excuse me, let me go back. And so what the algorithm attempts to do is it attempts to find a way of going through a graph, like is depicted on this set next to it, that attempts to step through each of those KMers and essentially aligning the KMers such that you would end up with a single path that takes you from one end of the genome to the other, representing all the different possible KMers that are there. Okay, and so that's fine, but and of course it's wonderful that we have computers to help us do that. But there's a couple of issues that arise and reasons that things might not work, and that are very important in keeping in thinking about your data. So this is a slightly different version of this, but it's really the same process. We're finding a path that steps through these KMers in an overlapping way to go through this graph. But what you can see in this particular case is, if, for example, in one of our sequences, we have an error. So the error is represented here by an A versus a C at a particular position in the sequence. And so we would have KMers that have the TGGA and TGGC. Well, instead of going through this path, now it forks and becomes 2 paths. And that A continues to represent an alternative version of this sequence. And if you're lucky, this would actually come back together and resolve itself so that you just had a bubble on your sequence. And so this is, if sequences were perfect and there was no variation, then this graphing method would be a lot easier. But one of the problems that comes up is that there is this property in sequences that when you're generating them, there are errors. And then in real organisms, there's often what we call polymorphism, so SNPs or actual natural variation that exists, and so those can become a problem and make this process very difficult. These are just some. If you think about it, what I've got here represented are some different paths or path problems that exist. And so the one that we just talked about, you could either produce a spur, meaning that you're able to align these KMers into a graph that has a single resolution, except for one spot of it where there's maybe an error or a polymorphism that produces what's called a spur. If a spur can get resolved, then you would have a bubble. are two alternatives through that path that have to be resolved because a contig will only have one sequence in it. And so algorithmically, you have to resolve this. And then the bottom one is a very important representation of part of the issue that takes place during assemblies, and is very important for us to keep in mind. So what is depicted below, and keep in mind that the thickness of the arrows have to do with how many KMers actually represent that path. And so you can see in the top two that those can be represented by an error, for example, that's only represented by one or a small number of KMers in the data set. And so it can be resolved because there is a dominant path through there. In the bottom one, it's kind of the opposite, where we're starting with two paths. So two contigs that are being formed. And yet in the middle, they join and become one and then separate again. And so what most likely gives rise to a pattern here, which is very difficult to deal with, is you can be making a contig from one part of a genome and a contig from another part of a genome and having unique paths that are taking place there. But then they start to share KMers because there's a sequence that's represented identically or nearly identically in different parts of the genome. And so this is a case where repetitive sequences, transposons or recent gene copies, all sorts of things that lead to the repetitive nature of genomes, really wreak havoc on assemblies. And so these things, unless they can be resolved by other information, end up breaking the assembly and making it so that your contigs are on average shorter. So, what's the concept of a KMer? So remember that KMers are the stretch or the length of DNA that you're going to use in your assembly. And so there's a couple of things that are important to keep in mind. That and a lot of modern algorithms for assembling sequences will actually run through different KMer sizes for you, and so eliminate this. In fact, the sequences that you're going to, the program that you're going to use to do your assembly actually attempts multiple different KMers and then picks an optimal range of KMers for your analysis. And so this is largely taken out of your hands, but it's important to think about it because if you think about it, this problem with having repetitive sequences, the repeat nature of a sequence depends upon length. So if I give you a sequence that's 10 nucleotides long, you may find many places in a genome where that 10 nucleotides would exist as a perfect copy of that 10. If I give you a sequence, on the other hand, that's 100 nucleotides long, well then that's much less likely to be represented. It would, of course, well be represented in many genomes multiple times, but it reduces the probability of having that sequence represented multiple times. And so in that sense, the larger the KMer, the better. The other problem with KMers is the longer they are, the more likely that KMer will include many different versions that have errors in them. And every one of those, it represents a KMer that will generate a spur and takes up computational space. And so there's a balance in terms of the KMer length that needs to be struck in terms of making for unique paths, which argues for making them longer, but not having so many opportunities to have random errors and polymorphisms that give rise to other problems for assembly. So once we have our assembly, Well, let's first of all, once we do our assembly, there's a couple of files that become very important. The one that's going to be most important to you is the Contig file. So this is the result of your assembly. So you've taken your sequences, you've passed them through your program, which in your case will be called SPADES. And that program will do its best to take all of your FASTQ files and assemble them into contigs. Very unlikely for any of you that you will end up, even if you started with a bacterium that has a single chromosome, with one FASTA file, one long contig. But you'll have a FASTA file or a multi-FASTA file, which means that it'll produce a file that has several to thousands of FASTA sequences that represent the original genome from your bacterium. And in some cases, you may also subsequently generate a SAM file by mapping your reads back to that Contig file. And so those are two very important outputs from that data that you'll see. The next thing that you'll want to consider, and this will be one of the steps in your analysis, a cost report that you produce here, is our way of understanding, when you do an assembly, talking about how good an assembly you have. And so there's a couple of ways that we talk about our assemblies that are very common. One measure of the quality of an assembly is what's called the N50. It's sort of a measure of average contig size. So the goal would be to produce a single contig that was whatever 5 million base pairs or whatever the genome size of your bacteria is. But in reality, that's not going to happen. But the N50 measure is specifically if you look at the entire assembly of your genome and you take your contigs and you rank them by size, right? What is the size of the contig when you've assembled half of the genome? So for example, if we have this 5 million base pair long bacterial genome and we do the assembly and then we rank the contigs by size. So you start with the largest one. Let's say the largest one is 2 million nucleotides long, or 100,000 or whatever. As we step through those, when we come to the contig where we've assembled 50% of the genome, that size, so let's say in this case, maybe it's 200,000 or 400,000 nucleotides, that tells you that half of the genome is now in chunks that are bigger than that, or contigs that are bigger than that. So that's a measure that's communicated often for the quality of an assembly. Another measure of quality is what's called the depth of coverage. So when you sequence your genome, of course, the idea is that every nucleotide would be represented in your assembly many, many times so that you get very accurate calls. But in reality, when we're looking at even if it's just a random representation of reads that come from the original sets of genomes that you broke up to start the process, there will be some regions of the genome that are not represented well. But what we try to do is to talk about the average coverage for a genome. And what that simply means is that on average, for all the nucleotides in your contigs, how many reads actually are represented or representative of that particular position, that nucleotide position on average. And so, for example, if you sequence a 5 million base pair genome and you have 10 million bases of sequence that you use to assemble it, then you'd have an average coverage of 2. And so if you have an average coverage of 2, you have every expectation that while some regions of the genome will have 10 sequences that represent them. Many others will have none. Whereas if you have an average coverage of 50 across the genome, you know that the vast majority of the genome will be represented by at least some reads. Another very important way of representing the completeness of an assembly is by looking at the gene catalog. And there are mechanisms for doing this. We probably won't do this in the context of your assemblies, but we might. One of the things that you can do is there are a set of genes in organisms that are known to exist in every single organism, even bacteria, basically core protein coding genes that exist. And so we can ask in our assembly, did I find all of those? Indicating that, yeah, we probably have the whole genome assembled here. It may be in chunks, but everything is there. Okay. And so that's just a principle that's used. Now, for the challenges that we will encounter, just to prepare you, we talked about these a little bit already. And this gets to the Humpty Dumpty problem, which is probably the most important. One is that there's heterozygosity and sequencing errors that exist. And so these are one of the major intrinsic challenges. And so you're trying to balance in these pathways for assembling contigs this problem that if you have too much heterozygosity, then you're going to be generating many different paths for a single representation of a chromosome. Okay, so one of the reasons that we're looking at bacteria is that because these are generally haploid, they only have one copy of each of their chromosomes. Sometimes they have multiple chromosomes. We hope to avoid that as a problem. The other is, even in bacteria, there's some level of paralogy, where there's recent gene duplications that exist. In us, there's this allelism, which gives rise to heterozygosity that we have to worry about. But even in bacteria, in some cases, there are going to be multiple copies of genes that interfere with the assembly. The most obvious ones that you'll come across are the ribosomal RNA copies. And so there are multiple copies of near identical ribosomal RNA genes in most bacteria. And so they are probably the single most problematic aspect of these genomes. And that's really also part of what is meant by sequence complexity, that the sequences are inherently repetitive, and those repeats can be as simple as little SSRs, so 2 nucleotides represented 100 times over and over again, but you don't usually find too many of those cases in bacteria, to large repeats like transposable elements or in fact the ribosomal RNA genes. And so from our Humpty Dumpty, you can see that when he broke, unfortunately, a lot of the fragments that Humpty broke into are essentially identical. And so exactly where those go is what leads to this idea that, all the king's horses and all the king's men are not going to be able to put him back together again. It's just an inherent impossibility. And should lead you to say, well, maybe we shouldn't break Humpty into so many pieces. It might be easier to put him back together. So the sort of extrinsic challenges to sequencing, some of them we've discussed already. One is the quality, and sequencing errors, of course, do not help with the assembly process at all, and if they're bad enough, lead to severe problems with putting things back together. The length of the sequence reads, the concept of repetitiveness is really related to sequence reads. So for example, we talked about technologies in an earlier video that produce really long sequences. And one of the wonderful things about those long sequences is that, if you have a sequence, if your sequences that you're trying to assemble are 10,000 nucleotides long, then then finding two sequences that come from different part of the genome that are 10,000 nucleotides long and so would be confused with each other is very rare. And so they really, when it comes to assembling complex genomes, they can be very, very helpful. Coverage, coverage is a random process in general, so there will be problems that there will always be regions of the genome, unless you spend a tremendous amount of time and effort to add sequence that will be undercovered. Sequences, because of the nature of the process, every one of those KMers, essentially unique ones, needs to be held in memory for most of these assembly algorithms. So it requires a lot of RAM, and so luckily you'll be doing your assemblies on a very high RAM server here that we maintain for you. And software, the software is really changing and improving remarkably over the last several years, but still remains somewhat a limitation in this. So thank you very much.